And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
I'll be speaking in English, en inglés. Lo siento, no hablo español ahora, pero en el futuro. I'd like to tell you about the process, the book I've written, Replacing Darwin. And I can't get through the entire book in 1 hour, which is why I wrote a book in the first place. So in the time we have remaining, what I'd like to do, instead of telling you the vast scope of what was in this book, instead tell you the process by which I came to my conclusions. So we'll go through 5 steps, and we'll get through some of the conclusions in the book along the way. Now, some of my background, I did my PhD research in adult stem cell biology. I went to graduate school wanting to work on cancer, and in 2003 the connection between cancer and stem cells was just growing, and that became my research thesis. I left that about 10 years ago to work on the question of the origin of species. Now I believe in following the evidence where it leads, and I've been surprised to see the health and origin of species connections come back together for reasons that you'll see momentarily. So, the first step, and this is to me applicable to any research topic, the first step in any research process is to find the question. I had many colleagues in graduate school who were good at doing experiments, good at producing results, but some still had trouble finding the question. This is an essential trait of any good researcher. Darwin defines the question for us. He says, the origin of species is that great mystery of mysteries. So that's the one I want to focus on. Now, for those of you who've done research, you know that the next step in this process is to rigorously decide what we know and figure out what we know and what we don't know. What's the unsolved problem? What I want to do is go back before Darwin and set up this context. So we talk about species because Linnaeus gave us the modern term, or something close to the modern term, in the 1700s. In his day, and for about 100 years thereafter, scientists believe that God created species in their current locations. They saw the match between species and their environments. It looked as if species had been designed for their environments, and they reached the simple conclusion that God had designed them, and they rejected the hypothesis that they were not designed. So if you were to look at the family tree of these species, it would just be long, unconnecting branches going backwards in time, never joining, back to the beginning. That's an explanation that's fairly simple to refute, and Darwin did a good job doing so. There's not that many observations you have to make to see evidence against this. And you can see some quotes here from his book. So Darwin came along after 100 years of God creating species in their current locations, corralled several lines of evidence against it, rejects that hypothesis, and embraces evolution. So that's the way things were in 1859. What I want to focus on now is what's changed, particularly among creationists, since then. And there's been 2 major changes. So that original view, as I've derived it, doesn't actually come from reading the Bible and taking it to nature. It just comes from making simple observations. I don't have time to derive it for you here. I've done it in print on many occasions. But modern creationists agree with Darwin in many respects, and they believe that the created unit is roughly approximated by the level of family. So if you look in Spanish Bibles, some of them say species. The Hebrew term is mean. The typo is typically the translation, the better, more appropriate translation. What I want to focus on is the implications for the origin of species. So under this modern creationist view, people like myself would believe that the species we see today are the descendants of ancestors that were divinely created, so that these species and these species and these species and all the breeds of dogs, as an example, they all belong to the same family, are the result of natural processes with time. In this general rule of thumb, we can illustrate with the family Equidae, that which includes the zebras and horses and donkeys and wild asses. All of these modern young earth creationists would say came from a common ancestor, in this case on board the ark about 4,500 years ago. So the contrast then is stark compared to 1859. Modern young earth creationists would agree that these species have a common ancestor. They would disagree on the time scale. So in this sense, it resets some of the debate. Many of the lines of evidence that Darwin used to disprove the fixity of species, the idea that God creates species in their current locations, modern young earth creationists agree with. So that resets it to both of these ideas trying to explain the same thing, the evidence fitting both. Now you might say, what about the other lines of evidence Darwin cited? After all, he's not arguing for the common ancestry of species within a family, he's arguing for universal common ancestry. So what about the evidence from homology? The shared anatomical structure, you can make the same argument in the embryological realm, you can make the same argument in the genetic realm. What about the existence of transitional forms like tiktaalik that seems to blend the features of aquatic and land creatures and seems to fill the gap between aquatic creatures and land ones? What about the structure of life? Basically from Linnaeus, the groups within groups or nested hierarchical classification of life that would seem to fit a process of descent with modification. Humans are grouped with primates because we share much more in common with them than we do with fish, but we're grouped with fish because we share more in common with them than we do with butterflies, and you have this groups within groups pattern, which would seem to fit this process of descent with modification over time, and the amount of similarity between any two species would seem to be a function of the time since their common ancestor. And like I said, you can see this as well at the genetic level, not just the anatomical. So wouldn't this seem to once again disprove even the modern creationist idea and get us no further than where we were in the 1859? Here's the second development among the creation community. The modern creationists have become better at recognizing the expectations of design. And again, I don't have time to derive in great detail where these expectations come from. But modern young earth creationists derive much inspiration from looking at the principles that human engineers invoke in creating things. So in this simple example, you have 3 vehicles manufactured by companies that are headquartered on 3 different continents, yet they all have an underlying structural pattern that they hold in common. Yet no one would believe that this underlying structural pattern was due to sharing a common ancestor in the Arctic and then developing over natural processes through time to see the features that we see today. My point is, humans naturally produce things in homologous patterns, and so this would also be consistent with a creationist hypothesis. Here's something that might surprise you and has surprised me in seeing how creationism has developed over time. I'm going to use a military analogy from the United States. This is a cruise ship, the Ticonderoga class cruise ship used for bringing firepower through the oceans. Here's an example of firepower that's used on land. This would be a terrible means of fighting in the oceans, and likewise, this would be a terrible means of fighting on land. What do you do if you want to move your firepower from the land through the oceans back to land again, or somehow go between those environments? You would design a transitional form that blends the features of ships and of tanks. It's inferior to a tank on land, it's inferior to a ship in the water, but it's superior to both for the transitional environment between water and land. So this would also be consistent with the hypothesis of design. Even the pattern in which life is organized, you find an echo in the human design realm. For whatever reason, the means of transportation that humans have designed naturally fall in a groups within groups, nested hierarchical pattern. You can see it at the visible level, you can see it at the blueprint level. So this nested hierarchical pattern of life would fit these hypotheses as well, both at the anatomical and genetic level. So my point here, and we could spend much more time on this, but I'm moving through it for sake of time, is that all the major lines of evidence for evolution also fit the design hypothesis. And this isn't because I'm trying to wiggle away, trying to find a way to fit creation. As I told you earlier, creationists believe in design and natural processes, and I'd love to have a tool that distinguishes between these two. I have a personal vested interest in finding that. What I've seen so far, have not found satisfying. So my point here is, that I've made very quickly, is that the question of the origin of species is still an unsolved problem. That's my point. Step 1, find the question, what is the unsolved problem? So all of these questions are still open from my vantage point. Now you might say at this point, as many of my opponents have done for over 40 years, especially in the United States, how is anything you've said science? Where are the testable predictions? Where are the predictions that future observations could reveal to be true or false? Well, we're only at step 1. So let's go to step 2. Find the unsolved problem. Now find the tools. This is something that's also surprised me in the 10 years of working on this question. For most of the last 150 years, we've argued about anatomical comparisons, about fossils, and other such things, and only recently has genetics been brought to bear on this debate. From my vantage point, that's the reverse of what it should be. It is that way for historical reasons, if you look at the history of molecular biology. But when sperm and egg meet, what's passed on directly, what directly records a species' ancestry and inheritance is DNA. Other fields of science are indirect, relevant, but indirect. So I would argue that DNA is the most important field of science on this question. And a second discovery that's been made in the history of modern genetics is that DNA changes with time. Mutations happen. So in a sense, DNA acts like a clock, recording the time since species first originated. And so by comparison of DNA sequences of various species around the globe, we can not only establish ancestry, but also time of origin. So these are the two major contributions of genetics to this question. So find the tools. What genetic tools do we have available to us? We have several thousand species with at least partial DNA sequences, and I'm thinking here of animals. We've got all sorts of bacterial sequences as well. My interest, though, has been in animals, and in particularly in humans. We've got over 40,000 human DNA sequences, either in part, thinking of mitochondrial DNA, or we've got increasing numbers of nuclear DNA sequences for humans. And you'll see, because humans are one of the best studied species at the genetic level, aside from many bacterial species, we often become a model then for the rest of life. Another piece of data that is numerically less than the actual genome sequences are parent-offspring mutation rates, both for mitochondrial DNA and for nuclear DNA. And just in case we have folks who are not necessarily geneticists, we're talking about in humans in particular, mitochondria here in orange, and the nucleus in purple. In humans, the mitochondrial DNA consists of only less than 17,000 base pairs or letters on average. The nuclear DNA is what most people think of when you get your DNA tested at one of these commercial places. That's primarily what they're looking at. And the nucleus contains billions of DNA letters. So these are the tools we have available. Third step in doing any sort of research project, the third step I've taken, is to study the familiar as much as possible. What is a great temptation in any scientific project is to jump to the hot question, to the big unsolved problem, when in fact the familiar fields, the familiar questions often have secrets that if you study them long enough that will be released. So let's start with one of those, one of the most fundamental facts of biology, the groups within groups pattern of life. I want to look at an example from mitochondria, and specifically from mitochondrial DNA and mitochondrial proteins that it encodes. You can see in mitochondrial molecular comparisons a groups within groups pattern. Here's an example from one of the papers I published. Along each of these axes, the y-axis and the x-axis, you have 2,700 animal species. So it's a highly zoomed out view of this. The red color represents sequences of high identity. The blue represents low identity. Thought I had my mouse, never mind. So anyway, you can see that the big red box there in the middle represents fish compared to fish. They're more similar to each other than to anything else, and they differ from other species. But it's in one of the papers I published. So what's the explanation for this? And let's press the argument further, as many of my opponents have done. This is a particular mitochondrial DNA protein. We know the biochemical functions that they perform in the electron transport chain. Take the ATP synthase as an example. From a design perspective, why would the zebra ATP synthase proteins have a different sequence from the toucan, as an example? This has been one of the arguments levied against the young Earth creationist view. From a design perspective, wouldn't you make them identical? And the fact that they're not, and in a groups within groups pattern, would seem to argue that they share that the differences reflect time since common ancestor. And in this case, we're looking at zebras and birds. This would fit the evolutionary idea, not the young Earth creationist one. Isn't this evidence for evolution? Well, is this the only function the ATP synthase performs? There's a little-known discovery that's been growing, and the numbers of these discoveries have been growing with time. It's this concept of protein multifunctionality. So here's some classic glycolysis enzymes, those enzymes involved in converting glucose to cellular energy currency. LDH there is lactate dehydrogenase. Well, you'd think these perform only one function in this particular pathway. They've also been found, lo and behold, in the nucleus, acting as transcription factors. This has been a very surprising discovery. So to make a long story short, the evolutionary argument I just presented to you implicitly assumes that the differences don't have a function. What I did in this paper, and I have it in English because that's what it was published in, what I did in this paper was derive from first principles, from a creationist perspective, a testable prediction on mitochondrial DNA function. And my prediction is that it's functional. The current data that we have do not resolve this question. It's a matter of future research. This is an example of something that future experiments could reveal to be true or false. So you start with the familiar, you study it to death in hopes that it'll yield some secrets. There's another element of mitochondrial biology that's relevant to our question. In a handful of species, the parent-offspring mutation rates have been measured, and the answer that we've seen from these studies has been consistent. So this is one of an early diagram from a paper that I published. The answer in each of these cases is the same. In red is the predicted number of evolutionary differences given the evolutionary time of origin. It's rather small, probably from where you're sitting. In blue, then, is the prediction from a young Earth creationist perspective, from the perspective that these species, or that humans originated only a few thousand years ago. And in green is the observed number of mitochondrial DNA differences that we see. And so in these four independent species, in each case, we see an overprediction from evolution and a young Earth prediction that matches what we see. And just to blow up here the human side of the equation, the modern evolutionary view puts the origin of humans, modern humans, about 200,000 to 300,000 years ago in Africa, and then about 50,000 to 100,000 years ago, humans come out of Africa and colonize the rest of the globe. We can look at the mitochondrial DNA differences among humans today, of which we have tens of thousands from... I'm just representing what the evolutionary literature says. So, here's the data that you can find in the evolutionary literature. It makes predictions. The question here in our case is, how long ago did humans originate? So if you apply the mitochondrial DNA mutation rate, which is about one mistake every 5 to 10 generations, and you do the math, what you find is the evolutionary model predicts, on average, about 1,500 DNA differences, a minimum of 477, and the number of DNA differences we see in Africans is about an average of 80. And in non-Africans, about 40, so you can see this is an over-prediction. If you do the same math with the young Earth model, which says humans originated from Adam and Eve about 6,000 years ago, that the people groups originated simultaneously at the Tower of Babel thereafter, what you find is if you run that same equation, it captures exactly what we see. And this is the type of data I put in the book. Now, what matters in the creation evolution discussion is testable predictions. Technically, this is a retrodiction, taking data that already exists, running the math, and see if it fits. A prediction would be to say, here's the rate we expect, and future experiments could show it to be true or false. So what I didn't tell you is that the rate of one mistake every 5 to 10 generations comes from measurements only in non-Africans. No one has yet measured the mitochondrial DNA mutation rate in Africans, and I put in print in the book a prediction as to what that would be. And my invitation to my evolutionary colleagues is to do the same thing, and then we can do an experiment and see which one is correct. So that's one element of step 3. I want to move on to step 4, which is follow the implications. And what I want to do now is tell you about another discovery, and then we can follow the implications from it. So this other discovery relates to mitochondrial DNA in animals. This is the phylogenetic tree based on mitochondrial DNA for species within the family Bovidae. We now have over 100 mitochondrial DNA sequences from these species. There's around 135 species total in this family. These branch points represent species split points. And if you graph out the pattern in which these species have arisen, you find that it's very linear. So that's the discovery. Now I want to tell you about these implications. You can find them more in this particular paper. Again, it's in English because that's what it was published in. I want to focus on these latter two implications. This one first. And I have to look at my English notes because I can't read Spanish. So one moment. There we go. I don't have time to derive the math in this particular case, but that linear rate implies that the rate of species formation per species is declining with time. Again, you'll have to see the paper for this explicit derivation. It implies that whatever is causing species to form seems to be losing its power or its ability to cause it with time. Okay, how do you explore that implication? We've been talking about mitochondrial DNA. Let's bring in nuclear DNA. And of the species that have mitochondrial DNA mutation rates measured, two of them also have nuclear DNA mutation rates measured. This is in animals. The water flea, Daphnia, and the fruit fly, Drosophila. What I'm showing you here, and I'm going to show you in the next slide, the fruit fly DNA differences. On the left, in blue, is the prediction for mitochondrial DNA differences. Black represents the actual differences. On the right, in orange, are the nuclear DNA predictions. Black is the actual number of differences. So what you can see in this slide is that the predicted number of mitochondrial DNA differences captures what we see, but the predicted number of nuclear DNA differences on a young Earth timescale severely underestimates what we see. And the same result holds true for fruit flies. Again, in blue, the mitochondrial prediction captures what we see. In orange, the nuclear DNA prediction underestimates it. And we have data as well for humans. So here, once again, is the mitochondrial DNA data in blue and a severe underprediction of nuclear DNA data there on the right. Now, both the evolutionary model and the creationist model have a contradiction to explain. For the evolutionary scenario, it's reversed. The nuclear DNA data matches, the mitochondrial DNA data does not. So both of these models must find a way to reconcile these two genetic compartments and make testable predictions from it. Here's how I've done it. Here's a diagram of the human DNA, the 46 chromosomes, 23 pairs. The hypothesis that I was resistant to initially, the way to explain these two DNA clocks, was to postulate pre-existing differences in nuclear DNA. In other words, I'm postulating that God created Adam with differences in his DNA. I found this hypothesis to be rather arbitrary initially, but it's grown on me, and in the book I describe 4 independent lines of evidence that point towards this. As it relates to our discussion about the implications of the mitochondrial DNA tree and the rates of speciation and the dilution of the number of species forming per time, this naturally explains that. If you have pre-existing DNA differences from the start, using basic, straightforward textbook population genetic principles, this leads to the dilution of those differences over time and to a decreasing rate of species forming on a per species rate. And once again, and I apologize in English, this is the paper in which you can find more detail where I drew these implications out. So that's one implication. I now want to focus on another one. If you take that graph that I showed you earlier and extrapolate it into the future, it implies that species will still be forming at a very high rate, which leads us then to step 5 in the research process. Find the question, find the tools, start with the familiar, and that rule has served me well for many reasons, and one of which I'll show you in a moment. Follow the implications and then rigorously question the results. This is what every good scientist does. And you can see on our website, we've invited people to review the book. We've got hostile reviews published there. I've done debates on this book online. You can find that as well. I debated Herman Mays, a PhD evolutionary biologist from Marshall University in West Virginia. If you just Google that, you'll find it there as well. And the best scientists learn to question their own results. So to give you a personal example, when I was going to graduate school, I was convinced of a particular explanation for what causes cancer. It was wrong. And it was a long process of figuring out that it was wrong. And I went in naively into graduate school thinking that this great idea that I was convinced was true. The only thing standing in the way were all these researchers who thought different. And in fact, as every good researcher knows, these people who oppose you are your greatest allies. Because at the end of the day, the greatest enemy of your hypothesis is not your competitors. It's reality. And you can have the best idea in the world and be wrong. And the quicker you know that's true, the faster you can move on to the correct hypotheses and move on. And so that was a critical but difficult lesson to learn. And so the best scientists learn to be their own worst critics. And that's what this step 5 refers to. In this book, I made other predictions, one of them being related to the, as I implied earlier, the rate at which species form. I want to tell you about a research finding that was published 4 months after my book came out. So this was published in October of 2017. 4 months later, a result in Darwin's Finches was published. But I want to set up the question for you here momentarily. There's around 11,000 bird species alive in the globe today. Here's how you could derive a prediction from the young Earth view. So as I mentioned earlier, young Earthers believe that species have descended from the kinds, the types that were on board the Ark of Noah. And so young Earth creationists would explain these 11,000 species by primarily natural processes. And I said that the young Earth creationists put the timing of the flood about 4,500 years ago. So you can then make a prediction about the average rate of speciation. 11,000 species divided by 4,500 years gives you an average rate of about 2 to 3 new species per year. There can be fluctuations within that. This is just a general average so we can move the discussion forward. And we can contrast that to the evolutionary view. And of course, birds would be the descendants of dinosaurs. Dinosaurs go extinct 65 million years ago. If you look at the evolutionary literature, modern birds would begin evolving around 35 million years ago. And so the average rate of speciation would be about 1 new species every 3,000 years. My point here is to show you that there's orders of magnitude difference in these two predictions, as you might expect, because the timescales are orders of magnitude different. Well, the finding in Darwin's finches was this. In 1981, a male of the large cactus finch, so that's in the box on the upper left, mated with several females of the medium ground finch, which is the box near the center. And one particular lineage, which the Peter and Rosemary Grant, the Princeton researchers who've been doing this for 40 years, they call this the big bird lineage. What they observed over time was that this particular bird species became reproductively isolated, genetically distinct, and morphologically distinct. It's got a different beak size. So everything that defines a new species has been identified here, and this was the paper in January 2018. What I want to focus on is the implications for the rate at which species form. So before this discovery, based on a 2015 paper in Nature, there were 18 recognized species of Darwin's finches. They were announced to the scientific community thanks to Darwin's voyage around the globe. He and Gould announced them in 1837, hence they bear his name. That's 181 years before 2018. So that's in the Galapagos, about 1 new species every 181 years. That's a simple, straightforward calculation. 1 divided by 100 is 0.01. 1 divided by 200 is 0.005. Both of those are a whole lot less than my predicted rate of 2 to 3 new species per year. However, I was making a prediction for the globe, not for the Galapagos. And so if you apply a conversion factor, convert the fact that we've been looking just at 18 species and apply that rate to the rest of the globe, divide by 18 times 11,000, what's the result that you get? So let's set these numbers in a row. The first number is 1 new species every 3,000 years. The second number is the number I just derived for you from a young Earth perspective. And the third number here is what you do if you go 1 new species divided by 181 years, divided by 18 times 11,000, you get 3.4 new bird species per year, which is exactly in line with what I've predicted. My point here is that the young Earth model is working. It's making predictions that future observations could reveal to be true or false. You can find more about that on our website, of course not in the book because it's a prediction. I have got a second article online that deals with the mechanism by which these species formed, which also fits the mechanism that I laid out in the book. I also want to tell you about research that's hopefully going to be published before the end of the year. This relates to another prediction I made in the book, and it relates specifically to human origins and to the Y-chromosome. I made little comment about it except to say that it should record the history of civilization if indeed human origins go back only a few thousand years instead of 200,000 to 300,000 years. So what we're looking at here is differences between fathers and sons, and then between all males going further back in history. So we looked at mitochondrial DNA, which I failed to mention is primarily maternally inherited, so that tells you female ancestry. This is male ancestry, the other side of the equation. So at the time I published my book, I was aware of only 2 papers that directly measured father-son mutation rates. This is the first one, looking at a Chinese pedigree. So two men who had a common ancestor in the 1800s. Then you measure the amount of Y chromosome differences between them, and the result was 3 times 10 to the minus 8 mutations per base pair per generation. The specific details aren't that important, just know that it's 10 to the minus 8th. We're going to look at that number again in a moment. I also want you to see that the sequence coverage, 12x to 20x, is fairly low. If you don't know what that is, it's basically a measure of the quality of DNA sequence. There was a second paper that looked at multiple Icelandic pedigrees and discovered the same answer, 3 times 10 to the minus 8th, and they had a similar level of sequence coverage or sequence quality. Well, what if you run those numbers through the equations that we did for mitochondrial DNA? What you see is this fits the evolutionary timescale, and of course, correspondingly, it strongly contradicts the young Earth one. Well, what's happened since then? There are 2 papers that have high-quality Y chromosome sequence that reported a different answer. And the pattern I've just shown you, which is low quality, low mutation rate, high quality, high mutation rate, is a well-known relationship in the evolutionary literature. Just look at the supplemental information for the Posnick 2016 1,000 genomes paper on male Y chromosome ancestry, 1,244 men. They describe that relationship exactly, and we see it played out here. This looked at 320 or so men around the globe. As a control for this experiment, they looked at 31 pedigrees, and they found a mutation rate that was about 10 times faster. You have to really dig into the supplemental information to find it. This paper in Nature from 2017 looked at 150 individuals, 50 parent-parent offspring trios. Again, it's high-quality sequence, and they found, again, a mutation rate that's about 10 times faster. So the pattern that we see is high-quality DNA sequences give you fast mutation rates. What implications does this have for the origin of the first man? You apply this again through the equations. As you might expect, the evolutionary model overpredicts this. The young Earth model captures exactly what we see. This leads to further testable predictions. Without deriving it, because it's basically textbook science, the Y chromosome phylogenetic tree allows you to infer changes in past population size. What am I talking about? So here's a diagram of changes in human population size over the last 3,000 years. The negative numbers refer to BC years, the positive numbers to AD. You can see in the last few hundred years there's been a spike in the human population growth. This is derived from archaeological records and historical records. Well, here's a Y chromosome tree derived from 171 men from around the globe, from Africa, Asia, Europe. You've got Native Americans in here, Australian Aborigines, Papua New Guineans, from around the planet. I don't have time to derive why the spot circled in blue is best supported by the evidence as being the root of the tree. My point is, if this is indeed only 4,500 years old, and you infer population growth from this tree, it should match the growth that we know from the last 3,000 years. This diagram shows you exactly that match. The 2 black lines represent the statistical variability in the estimates of past population size. Of course, there's going to be variety because it's based on archaeology and historical records. The gray lines represent the statistical variety inferred from the Y chromosome, and you can see there's about a 90% match. So yet another testable prediction that's coming true, and this opens up a whole new research area, which if we had more time, we could go through in great detail. Now, let me deal with one potential objection to the research. Much of this is published in creationist journals, not in the mainstream literature. And this is often an objection I hear from my opponents, from people who are wondering about the research. Why aren't you publishing in Nature or Science or Cell or one of these major journals? One of the responses I give and the illustrations I give comes from the study of human DNA function. So when the human genome was published in 2001, there were many announcements that most of it was filled with junk, basically. Well, about 11 years later, in 2012, the ENCODE project did biochemical experiments on all of our billions of DNA letters, and based on biochemistry, argued that 80% had preliminary evidence for function. It's an ongoing project. In the peer-reviewed literature, you can find articles that criticize this. So this is one of the reasons for the criticism is that this high level of functionality is not consistent with the expectations of evolution. So here's a paper by Dan Grauer and colleagues arguing that it must be wrong because it disagrees with evolution. My point in bringing this up is the following. When people say, why don't you publish in the evolutionary literature, in the mainstream literature, they're assuming that a hypothesis that questions evolution would be published. What I want you to see is that the mainstream literature does not treat evolution as a hypothesis to be tested. It treats evolution as the test of other hypotheses. So by definition, it would be impossible to publish it there, and that objection is fundamentally a circular argument. Now, my book focuses primarily on biology. You won't see much discussion of geology, astronomy, because I'm speaking only to my discipline and that which I know. I refer to the research by my colleagues in geology and astronomy. They can have that debate there. My focus, again, is on the question I think is important and that I can personally address, namely the origin of species. I've set up a Facebook page for the purpose of engaging and having dialogue. I realize this is a contentious issue. I don't think you'll find the book to be polemical. My goal in writing this book was to show the research, and the reason I said replacing Darwin instead of replaced, because science never stops. As you can see, the research is ongoing. I've got projects in the works, and this is an ongoing dialogue, so replacing Darwin is the Facebook page you can find. To have this dialogue, I've also got my personal Facebook page, Nathaniel Jeanson. The website where you can find some of the research that I referred to earlier is the English one, answersingenesis.org. The Spanish version is Respuestas en Génesis. Thank you so much for your attention.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button, because we are just getting started.